The immune system is the body's defense system. Our body is under constant attack from all kinds of microorganisms. Most of the time we remain healthy because our immune system recognizes foreign material and destroys it, protects us from it. The word immunity means to be free from disease. The immune system can be divided into three lines of defense. The innate defense system is nonspecific. The first line of defense are the physical barriers of your body, and the second line of defense are the chemical barriers of your body. Everyone has pretty much the same first and second line of defense. The adaptive or specific defense system is unique for each person. This third line of defense is defense against particular foreign substances. It will vary from person to person because it will depend upon what you have come in contact with. Because the third line of defense has to develop an attack system for a specific thing, it takes a little while to activate this system. The immune system is a functional system. Most systems are a collection of organs that all work together for a specific function, but the immune system borrows bits and pieces from other systems. In the immune system, we have a diverse array of molecules and cells, as well as bits and pieces of lymphoid tissue that act as surveillance sites for these cells. The innate and adaptive systems are not separate, but rather intertwined. Both release and recognize many of the same defensive molecules, and our innate responses are not as nonspecific as we once thought. Many of the proteins released during the innate responses alert the adaptive system, and this makes sense since the adaptive system is going to take a while to come into play. If we have our first and second line of defense being breached, our third line of defense needs to be ready to go. Here is a little summary of your innate defenses, the surface barriers, and the internal chemicals. And then your adaptive defenses, which will be your humoral immunity, antibodies, and your cell-mediated immunity. We'll talk about each of these. The first line of defense is your intact barriers. The first line alone may be enough to ward off most pathogens. Barriers keep any kind of invader out. The first line of defense will be your intact skin and mucous membranes. You'll remember that between the skin cells there are tight junctions. These are like Ziploc bags and are designed to keep things from getting between the cells. Also, your skin has the protein keratin in it, which makes the skin waterproof. All body cavities with an opening to the outside is lined with a mucous membrane. Cavities that are open to the outside are ways that microorganisms can enter the body. These mucous membranes have mucus on them, as well as protective chemicals. The protective chemicals of the skin and mucous membranes include things like the acids that are naturally found on the skin, in the vagina, and the stomach. Acids inhibit bacterial growth. There are a number of enzymes that are secreted in various body secretions. A common one is lysozyme. Lysozyme is found in the saliva, in the respiratory mucus, and in the lacrimal fluid. This is why people can take their contact lenses out, clean them off in their mouth, which is one of the germiest places on earth, and pop them back in their eyes and not have their eyes rot out. Those tears take care of a lot of the bacteria. Mucin is a protein that when it's dissolved in water makes this really thick, sticky mucus. And mucin is something that lines the respiratory and digestive tract. This thick, sticky mucus acts to trap bacteria. Other cells may then help move the mucus with the trapped bacteria to natural openings of the body. Defensins are a group of antimicrobial peptides that are nonspecific in nature. They increase in output whenever inflammation starts. This would be anytime tissues are breached. Some other chemicals that are toxic to bacteria include the oils and sebum, and a substance called dermacidin, which is found in sweat. The second line of defense are the cells and chemicals that are under the barriers. They come into play if this first line is breached, if the barriers are broken. Your second line of defense includes phagocytes, natural killer cells, the process of inflammation, antimicrobial proteins, particularly complement and interferons, and fever. Your second line of defense acts based upon some pattern recognition receptors. These are nonspecific kinds of proteins that are found on cells that are not part of your body. In other words, this is not a particular pattern that's specific, it's simply something that's not normal to the body. The phagocytic cells include the neutrophils and the macrophages. The neutrophils, you'll remember, are the most abundant white cells in the blood. They become phagocytic upon encountering any kind of infectious materials in the blood or body tissues. Macrophages are monocytes that leave the blood. These are some of the most active phagocytes in your body. Free macrophages wander through the tissues of your body, coming and going from the blood. But some tissues have permanent resident macrophages that are always present in the tissue. For example, the stellate macrophages in the liver are part of the liver cells. Phagocytosis starts with the ingestion of a particle by the process known as endocytosis. A vacuole forms around the ingested particle, and this is known as the phagosome. The phagosome will merge with a lysosome and may be called a phagolysosome, and the enzymes of the lysosome will digest the material in the phagosome. Generally, this kills the ingested material. However, some pathogens are resistant to lysis, the organism that causes tuberculosis, for example, and some intracellular parasites. These organisms may even multiply in the phagocyte and sort of use the phagocyte as a place to hide out from the immune system. If we look at the steps of phagocytosis, the phagocyte will adhere to the particle it's going to ingest and form a phagosome. The lysosome will then fuse with the phagosome, releasing the digestive enzymes, and the material will be digested. Typically, this is what kills it. And sometimes exocytosis occurs as there are undigested particles that are eliminated from the cell. Here you see one of those phagocytic cells in an electron micrograph actually working, taking in bacterial cells that it's going to kill. Sometimes cells are not digested by the lysosomal enzymes. Then the cell may undergo something called a respiratory burst. Chemicals from other immune cells will stimulate the macrophage to produce additional enzymes. Some of these enzymes cause liberation of highly destructive free radicals like superoxide. Others will cause the cell to produce oxidizing chemicals like hydrogen peroxide and even bleach. Also, the pH of the phagolysosome may increase, as may the osmolarity. Combining all of these things together typically kills whatever is in the phagolysosome. The first thing that has to happen in phagocytosis is the macrophage has to attach to the particle it's going to ingest. If bacterial cells are covered with a capsule, this may cover their attachment proteins. Some bacteria have a rather slimy capsule that covers them, making them hard for the phagocyte to attach to. The bacterial cell may be then coated with special proteins called opsonins. This is a process called opsonization. These opsonins provide attachment sites for the phagocytes. It allows the phagocytes to now ingest the cell. Natural killer cells are a population of large granular lymphocytes. Most lymphocytes become educated. They learn to be T cells or B cells, but this small population of lymphocytes do not get that education. These cells will lyse and kill cancer cells and cells that are infected with a virus. Cancer cells and virally infected cells produce abnormal proteins, proteins that are not self. Natural killer cells are not programmed to look for a particular not self protein, just recognize any protein that's not self. Because the natural killer cells are not phagocytic, they kill by direct contact. That is, they attach themselves to the offending cell and cause it to lyse. They also can secrete chemicals that enhance inflammation, causing other cells to be called to the area to assist with destroying the cell. Inflammation is a response to any damaged tissue. Some people confuse inflammation with infection, but physical trauma, intense heat, or irritating chemicals can damage tissue, and that will cause inflammation, as will an infection. Inflammation helps to prevent the spread of damaging agents to nearby tissues. It helps dispose of cellular debris and pathogens. It also plays a role in alerting the adaptive immune system. We may need some specific immunity to help protect us from the invader. It also sets the stage for repair. The cardinal signs of inflammation are redness, heat, swelling, and pain. And sometimes a fifth sign is impaired function. If the area is swollen or painful, you may not want to use it as much. This allows for the area to rest, which may be a key thing to helping it repair. Inflammation starts because inflammatory chemicals are released. The mast cells released histamine, which is the primary chemical alarm. But other damaged cells can make and release a host of inflammatory chemicals. Macrophages and certain boundary cells are cells that are found on the edges of your tissues, like under the skin and under the mucous membranes, also have special pattern recognition receptors. These toll-like receptors recognize particular groups of invading organisms, not a specific organism, but a group. For example, some toll-like receptors recognize gram-negative bacteria. This will trigger a release of certain cytokines that are effective against that group of organisms. Kinins, prostaglandins, and complement are also released by damaged tissue. All of these chemicals have many of the same functions. They dilate local arterioles. This makes the blood rush into the area. They also make the capillaries leakier. They make the capillaries swell, opening up those intercellular clefts, allowing more fluid to leave the blood and get into the tissues. And leukocytes are attracted to the area. Vasodilation counts for two of the cardinal signs, redness and heat. If you open up the blood vessels, then hyperemia results. Hyperemia is excessive blood in an area. The area becomes congested with blood, and blood is both warm and red. The inflammatory chemicals also increase the permeability of the capillaries. This means that fluid escapes from the blood into the tissues. This is referred to as exudate, and it will cause edema or swelling in the area, another of the cardinal signs of inflammation. The swelling will place pressure on nerve endings, and this will account for the pain. Edema is this surge of protein-rich fluids. One of the things that happens is as the fluid washes through the area, it sweeps the area of debris. You will remember that the lymphatic vessels are capable of taking in particles as well as fluid, so those particles will then be taken or removed from the tissue. Also, these fluids deliver complement and clotting factors to the area. Complement is important in warding off invading organisms, and the clotting factors will cause fibrin to form. This will wall off the area and help prevent the spread of damage. Phagocytes have to be called to the area. Phagocyte mobilization occurs when phagocytes leave the blood. The first cells to leave are the neutrophils, and then the monocytes follow. If bacteria are involved in the inflammatory process, complement is activated and various elements of the adaptive immune system come to the site, like lymphocytes and antibodies. Phagocyte mobilization is a four-stage process. It starts with leukocytosis, then there's margination, diapedesis, and chemotaxis. Leukocytosis occurs because the injured cells release leukocytosis-inducing factors. This causes white blood cells to be released from the bone marrow and increases the white cell count in the blood. While these white blood cells are floating everywhere in the blood, we want to make sure they go to a certain place. This is why margination is important. The inflamed endothelial cells develop cell adhesive molecules, or CAMs. These sort of act like Velcro, and as the white blood cells are flowing by, they're stuck to the edges of the capillary. Once the blood cells are stuck to the edges of the capillary, they will simply move through those intracellular clefts, that process of diapedesis. And then because the inflammatory chemicals are specific for white blood cells, the white blood cells will be able to find the exact point of inflammation and migrate there. That's the process of chemotaxis. So here we see our injury, and we have a lot of white blood cells entering the area. The endothelial cells produce CAMs, So margination occurs, and once the white blood cells are stuck to the endothelial wall, they will move through it by diapedesis and be called specifically to the area of injury by chemotaxis. In severely infected areas, pus typically forms. Pus is simply a mixture of dead or dying neutrophils, tissue cells, and living and dead pathogens. Normally, the inflammatory process clears this area, but if it fails to, collagen fibers will form to wall off the area, and you will have an abscess, a pocket of pus. Some bacteria may be trapped in this pocket of pus, resisting digestion by the phagocytic cells. These organisms can live in the macrophages, and this is how they escape the effects of antibiotics and hide from other aspects of the immune system. If this happens, these long-term areas become granulomas. In the granuloma, the bacterial cell may still be alive, but the person is symptom-free. As long as the granuloma stays intact, there's not a problem. But if the granuloma breaks down, then the organism is free in the system again and can cause disease. So if we look at the overall thing from tissue injury, the injured tissues will release all of those inflammatory chemicals. We'll have the arterioles dilate with local hyperemia that will give you heat and redness. This will locally increase the temperature, which will increase metabolic rate of the cells, helping with healing. The increased capillary permeability will cause the capillaries to leak that fluid, exudate will form. We'll have this edema, the swelling in the tissues and the pain, and maybe some temporary impairment of function. Again, that leads to healing. The inflammatory chemicals also attract neutrophils, monocytes, and lymphocytes to the area by chemotaxis. These leukocytes may migrate to the injured area. Margination occurs, so they cling to the capillary walls. We have diapedesis, phagocytosis, and the area is cleared of debris. Healing occurs. Notice that pus may form there. Because of tissue injury, those leukocytosis-inducing factors is another one of the contributing factors that brings white blood cells to the bloodstream, again, so that the leukocytes marginate, go through the capillary walls by diapedesis, and are involved in phagocytosis. The end product of the inflammatory process is to have a healed tissue. Antimicrobial proteins enhance innate defenses. They either attack microbes directly or they hinder their ability to reproduce. The two most important antimicrobial proteins are the interferons and the complement proteins. Interferons mostly act against viral infections. When viruses invade a cell, they take over the metabolism of the cell to make new virus particles. Once the new virus particles are made, the infected cell is lysed, virus particles are released and can infect nearby healthy cells. The infected cells can secrete interferons. They can't save themselves, but they can protect nearby cells. These interferons will diffuse from the infected cell to nearby cells. And in nearby cells, they'll stimulate the production of a protein that interferes with viral replication. Even though the virus may infect the healthy cell will not be harmed because it will block viral replication. Interferons are not specific for a particular virus, just viruses in general. So here you see a virus infecting a cell, taking over the host mechanisms, making more virus particles which will be released from the cell. But this viral infection also has caused this infected cell to produce interferons. And interferons diffuse to nearby cells. When they attach to the receptor for interferon, they trigger chemical changes in the healthy cell so that it makes this interfering protein. Even though the virus comes in, it can't do anything, which means this cell stays healthy and stays uninfected. Interferons are a whole family of immune modulating proteins. Alpha and beta interferon are primarily antiviral. They can also activate natural killer cells, so they can play a role in cancer. Gamma interferon is secreted primarily by lymphocytes. It has a widespread effect on the immune system, mobilizing a number of cells. It also activates macrophages. Since macrophages and natural killer cells can act directly against cancer cells, interferons may play an indirect role in battling cancer. Interferons have been used to treat a number of diseases, including hepatitis C, genital warts, and multiple sclerosis. Complement is another one of the nonspecific proteins. It's actually a group of 20 plasma proteins. They circulate in the blood in an inactive state, much like your coagulation proteins. Their major mechanism is to destroy foreign cells. When these proteins are activated, it's the same kind of cascade effect we saw with the coagulation factors. Activated complement can play several roles. It can release inflammatory chemicals that amplify all aspects of the inflammatory process, but its primary role is to lyse and kill certain bacteria and cells. There are three ways that we can activate complement. The classical pathway involves an antigen-antibody complex forming. If we have an antigen-antibody complex, that means that the adaptive immune system is already in play. This antigen-antibody complex binds complement, and binding complement is the first step in the pathway. The lectin pathway involves a water-soluble protein that's produced by the innate immune system. It's produced in response to foreign material. Lectins can bind to sugars on bacteria and thus fix complement, starting again the complement cascade. In the alternative pathway, C3, which is the first of the complement proteins, is activated directly by microorganisms. Complement can act as an opsonin, so it can enhance phagocytosis. Complement can also amplify the immune response by triggering histamine released from mast cells, which would stimulate inflammation, and by attracting neutrophils to the area. But its real showstopper is lysing a cell. All the complement components, when they come together, they form a membrane attack complex, a MAC. And what this membrane attack complex does is it punches a hole in the membrane of the cell, killing the cell. So as we look at the pathways, three pathways, all of them activating C3, which is the first of the proteins that has to be activated. Now right off the bat, one of these C3 activations acts as an opsonin, so we can have enhanced phagocytosis. Another one, plus this one down here, can enhance inflammation. It stimulates histamine release, increases blood vessel permeability, attracts phagocytes, that whole 9 yards there. But if we get the whole scheme done, we lay down this nice little hole punch that literally pokes a hole in the membrane of the offending cell, and that's going to kill the cell. The last of our innate techniques is fever. Fever is any time you have an abnormally high body temperature. Leukocytes, macrophages, and certain other cells can release chemicals called pyrogens, and pyrogens sneak up to your hypothalamus and reset your body temperature, sets it for a higher temperature. Your body goes through all of the necessary things to make yourself get warmer. Fever is an adaptive response that benefits the body. At one time we thought it was just one of those annoying side effects of disease, but it turns out it's really a good thing. One of the things high temperature does is it causes the liver and the spleen to sequester iron and zinc. Many bacterial cells require iron and zinc to divide. If they have the maximum amount of iron and zinc, then they can divide at their maximum rate. But if the iron and zinc is limited, it will limit their bacterial growth. We can't take enough iron and zinc out of the system to stop them from dividing, but we can certainly slow down the process, and that gives your immune system a chance to get ahead. Also, fever increases the metabolic rate of tissues. This should speed repair. And it generally makes you feel bad so that you rest, an important thing if you're trying to get over something.